லேயரா போட சொல்லிருக்கோம்ல சார், பண்ண சொல்லிடலாம்.
என்ன பண்ணலாங்க?
ஜாயின் ஆகட்டும் சார்.
இல்ல லைவ் ஸ்ட்ரீமிங் எல்லாம் ஓகேவா, அதுக்கப்புறம் பண்ணிக்கலாமான்னு கேட்டேன்.
10:50 லைவ் ஸ்ட்ரீமிங் இது பண்ணலாம்னு சொன்னாரு எம்மி நேத்து.
லைவ் ஸ்ட்ரீம் போட்டார் போகுது.
அதான் டைம் செட் ஆனது அந்த டைம் தானே நேத்து சொல்லிட்டு இருந்தாருங்க.
பிகே நான் லைவ் ஸ்ட்ரீம் போட்டேன், யூடியூப்ல வந்துருச்சு.
ஓகே, இப்போ வந்து நம்ம அட்மிட் பண்ணிட்டு அப்படியே ஸ்டார்ட் பண்ணா சரியா இருக்கும். இனிமேல் நீங்க பேசுறது வந்துட்டு இனி வந்து பேசும்போது பார்த்து பேசுங்க. எல்லா பார்ட்டிசிபென்ட்ஸுமே உள்ள வந்துருவாங்க.
ஆமா, நாம வந்து அவங்கள வீடியோ ஆடியோ ரெண்டுமே மியூட் பண்ணிட்டு தான் உள்ள அட்மிட் பண்றோம்.
ஓகே, இப்போ அட்மிட் கொடுத்துடலாமா?
கொடுத்துடலாங்க.
ஓகே,
எல்லாரோடதுமே மியூட் பண்ணிட்டு வீடியோ எல்லாம் எதுவுமே பண்ண முடியாது. அவங்க
இப்போ பார்ட்டிசிபென்ட்ஸ் எல்லாமே உள்ள வந்துட்டு இருக்காங்க மேடம், நீங்க அதுக்கு தகுந்த மாதிரி பேசி.
அந்த வெயிட்டிங் ரூமே நான் எடுத்துட்டேன், வந்தா அவங்க பாட்டு வரலாம் உள்ள.
ஓகே ஓகே, ஆட்டோமேட்டிக்கா அட்மிட் ஆயிடும்.
ஆட்டோமேட்டிக். ஆமா, ஆட் பண்ணுவாங்க.
இப்போ உங்க அங்கிள்கிட்ட சொல்லிடுங்க.
எம்மி சார்.
மேம் சொல்லுங்க மேம்.
சார் நேத்து சொன்ன மாதிரி இப்போ ஸ்டார்டிங்ல எல்லாருமே வந்துட்டு வீடியோ ஆடியோ வந்து மியூட் பண்ணிக்கலாம் இப்போ பார்ட்டிசிபென்ட்டோடையத. அதுக்கப்புறம் ப்ரோக்ராம் கரெக்டா ரன் ஆயிட்டு இருக்கிற டைம்ல டிஸ்கஷன் டைம்ல வந்துட்டு அந்த மியூட் ஆப்ஷனை இது பண்ணி விட்டுருங்க, ரிமூவ் பண்ணிடுங்க. யார் வேணாலும் மியூட் பண்ணிக்கிற மாதிரி, அன்மியூட் பண்ணிக்கிற மாதிரி.
ஓகே,
பிரசன்டேஷன் முடிஞ்ச உடனே. ஆமா, பிரசன்டேஷன் முடிஞ்ச உடனே. ஆமா,
இல்லாட்டி அதுக்கப்புறம் ஹேண்ட் ரைஸ் பண்ற மாதிரி இருக்கும். அதுக்கப்புறம் யாருன்னு பார்த்து நம்ம எடுக்கிற மாதிரி இருக்கும்.
ஆமா,
அதுதான் கரெக்டா. அத இது பிரசன்டேஷன் முடிஞ்ச உடனே அது மறக்காம இது பண்ணிடுங்க.
ஓகே
ஓகே சார்.
ஹோஸ்ட் பண்ணனும். சாரி மேடம், பிரின்சிபால் மேடத்துக்கு லிங்க் அனுப்பாம விட்டேன், அவங்களே போன் பண்ணிட்டாங்க. லிங்க் அனுப்பிட்டேன். இப்ப அவங்க வந்தாங்கன்னா அவங்க ஹோஸ்ட் பண்ணணும். அதான் அவங்க வேற யாருனாலும் பாருங்க, மேடம் வந்திருக்காங்களான்னு பார்த்து அவங்கள ஹோஸ்ட் பண்ணிடலாம். மேடம் பிரின்சிபால் மேடம் வந்திருக்காங்களான்னு கொஞ்சம் செக் பண்ணுங்க, அவங்கள ஹோஸ்ட் பண்ணணும். இப்பதான் லிங்க் நான் காலையில அனுப்புறேன்.
வந்திருக்காங்க சார், அவங்க மேடம் இருக்காங்க.
Principal madam, good morning madam.
Madam Principal, good morning.
Okay.
மேடம் மியூட்ல போட்டிருக்காங்கன்னு நினைக்கிறேன் மேம்.
யாரு பிரின்சிபலா?
மேடம் பிரின்சிபல் மேடம், பிரின்சிபல் மேடம்.
Sorry, நான் அன்மியூட் பண்ணலையா? Okay, sorry. Good morning.
ஓகே மேம்.
Good morning.
குட் மார்னிங் மேம்.
Good morning.
Good morning, ma'am.
Good morning to all.
Resource person has also come.
I can do it now.
Yes. Welcome,
Yes, sir.
sir. Good morning. Good evening.
வெல்கம் சார். குட் மார்னிங்.
Sir,
Hello.
welcome. Good morning.
Good morning.
Good evening. Good morning.
கேக்குதுங்களா நான் பேசுறது?
கேக்குதுங்க சார்.
Okay, all right, cool. All right. சாட்டர்டே உங்களெல்லாம் செமினார் கூப்பிட்டார் போல எல்லாம் எழுதி.
Yes, we have to thank you, sir. You are a
It's okay.
yeah.
That's okay. It's no problem.
Thank you, sir, for accepting.
சரிங்க.
Start பண்ணலாமா?
பண்ணலாங்க. நீங்க ரெடினா நான் பண்ணலாம். ஒன்றும் ப்ராப்ளம் இல்லை.
ஸ்டார்ட் பண்ணலாமா மேம்?
Sir, PK sir, ME sir.
67 பார்ட்டிசிபென்ட் தான் இருக்காங்க.
Can we wait 10 more minutes, madam? Only 67 participants there.
Okay.
Yeah, we can give another 5 minutes. Yeah.
Yes.
5-6 minutes. Yeah. Last time ஊருக்கு வரும்போது அகிலா மேடம் வீடு தான் வந்து பார்க்கலான்னு பார்த்தோம். டைம் இல்லை.
Sir, I have really missed you, sir. அது உங்களுக்கு ஒரு ஷார்ட் டியூரேஷன்ல.
ஆமா.
Actually நாங்க வந்து much years later நீங்க அங்க ஜாயின் பண்ணிருக்கீங்க. CASல.
ஆமா.
ஜாயின் பண்ணிருக்கீங்க.
நான் 88ல ஜாயின் பண்ணேன். மாதவன் லேப்ல.
88 தான் நீங்க ஜாயின் பண்ணிருக்கீங்க. I thought நீங்க 86 ன்னு. Okay.
ஆமா. 88 எண்ட்ல ஜாயின் பண்ணேன். உங்களுக்கு சிவசாமி சார் உங்களுக்கு ஜூனியருங்களா?
இல்ல நான் வந்து ப்ரொஃபசர் ராஜாராவ் கூட இருந்தேன் சார் அங்க அந்த லேப்.
ஓகே.
அங்க அதுல இருந்து சார். நான் இங்க கீழ வந்து நடராஜன் சார். நான் அவரும் both of us got selected for இது நம்ம பச்சைப்பஸ் காலேஜ். அவர் வந்து ஸ்டூடெண்டா.
சந்தோஷம்.
86 later half of 86 I joined service. நீங்க அப்ப ஸ்டூடெண்டா இருந்திருக்கீங்க.
86ல வந்து நான் பிஎஸ்சி முடிச்சிட்டு 86 to 88 வந்து எம்எஸ்சி முடிச்சேன் நானு.
ஸ்டூடெண்டா இருந்திருக்கீங்க.
ஆமா, அப்பதான் ஸ்டூடெண்ட் நானு. அப்போ முடிச்சுட்டு மகாதேவன் லேப்ல வந்து லேட் ஒரு ப்ராஜெக்ட்ல ஜாயின் பண்ணேன்.
ஓ ஓ அப்படி வந்தா ஓகே ஓகே
நான் எம்ஃபில் போல I got directly into
ஓகே. எங்க டைம்ல வந்து எம்ஃபில் இது மஸ்ட்.
ரைட். நான் ஆக்சுவலா எம்ஃபிலுக்கு போயிட்டு கிடைக்கல. அப்புறம் ஒரு ப்ராஜெக்ட் இருந்தது, அப்ளை பண்ண உடனே அதுல கிடைச்சது.
ஓ சரிங்க. ஓகே. But last time வந்து we very much wanted to have your talk, but time was very very short and then we could not arrange அதுல.
லாஸ்ட் டைம் சொன்னாரு, இளவழி சொன்னாரு, எங்க காலேஜ் வந்து கொடுங்க மாமான்னாரு. அப்போ எனக்கு கொஞ்சம் அலைச்சலா இருந்தது, வர முடியாம போச்சு. அப்போ
பட் லாஸ்ட் வந்து நீங்க கொடுக்குற மாதிரி இருந்துச்சு. then we could not arrange here. Probably 22
பார்க்கலாம். 22 நம்ம it's God's grace. If there is opportunity to come, we'll come there.
Very much we would like that.
ஏன்னா லாஸ்ட் இயர்
14ல நான் அங்க இருந்தாங்க.
சந்தோஷம்.
14ல நான் அங்க இருக்கிறது வந்து I was talking to அவங்க உங்க வைஃப் கூட பேசிட்டு இருந்தாங்க. either almost 1 week அங்க இருக்கும்போது ஃபுல்லா நாங்க பேசிட்டு. actually she was to meet me at Dublin.
Okay.
2 hours drive or some 1 hour drive இருந்துச்சுங்க.
ரைட். நாங்க அப்போ சான் ஹோசேல இருந்தோம். ஆமா, அப்போதான் ஆக்சுவலா வந்து we moved back from New Jersey to Bay Area 2014ல.
Yes.
Yeah. வாங்க, next time வாங்க, கண்டிப்பா will meet and also will take you around little bit.
Carolina, your daughter, she has done university, sir.
இப்போ சீனியர் போறா, இப்போ UCLA-ல இருக்கா இப்போ.
Okay.
She is also studying biology.
Okay. Yeah, I worked the details from. Sir, how many of them have joined now?
122, madam.
Oh, wow.
Start madam. 122.
You can start.
You can start, madam.
Start pan.
Palla, you can start.
Okay.
You can start.
Principal madam சொல்லுங்க.
Yes.
Inform the principal madam.
Good morning.
Good morning, sir.
Good morning, madam. Start
Good
பண்ணலாம்
morning,
madam.
sir. Good morning, sir.
Good morning.
Good morning to all. I'm start பண்றேன் ma'am.
Yes, ma'am.
Sir, start பண்றேன்.
Yes, ma'am.
On this beautiful morning, we are pleased to welcome each and every one of you for this brainstorming webinar. I want to express my heartfelt thanks to God Almighty. I know without Him nothing is possible. I also think and I also pray Pachaiyappa to shower His blessings. On this special occasion, I, Dr. J. Kavina, Assistant Professor, Department of Botany, welcome you all for this brainstorming webinar on the topic recent development in genomic medicine. It is my privilege to invite HOD Madam Dr. V.M. Akila to give a welcome address. Please, ma'am.
Thank you, Kavina. Good morning and greetings to all. Respected principal, person of the department of our college, faculty and research colleagues from various colleges and various labs, and my dear students. Welcome to you all. As we enjoy the thought, me and Dr. Edumalai, we belong to the same school of study. I recollect my olden days at Botany, at Agiti. Currently, Dr. Edumalai is design and development of microarrays for different crop plants. Sir, with your permission, I just would like to say to my students what a microarray is, because currently this semester is the cell biology and genetics. Students, microarray is it's a lab tool that helps to detect the expression of thousands of genes at the same time. And Dr. Edgar is currently working on a curriculum, and he has developed a curriculum for the scientists working on genomic product line. Sir, across the nation, people are waiting to listen to your informative talk. On behalf of everybody of Pachaiyappa College, we welcome you to this webinar organized by our faculty members of Botany Department, Pachaiyappa College. So I welcome the principal and everybody and our speaker for this day. Thank you so much for accepting our invitation, sir. Thank you so much. And now over to Kavina.
Thank you, madam. Thank you very much for your mind-blowing welcome address. May I now invite our principal madam, Dr. Sri Jayanti, to give presidential address. Please, ma'am.
Yes, respected head of the Department of Botany, respected resource person, Dr. Rangasamy Erumalai, dear colleagues in the Department of Botany, students and participants of this webinar. A very good morning to one and all present here. It's a great honor to me to preside over this webinar. The Postgraduate and Research Department of Botany, Pachaiyappa College, is one of the pioneers in imparting high quality education, undertaking research of eminence, and consistently creating innovations in the field of botany and related sciences. Being an organ of the 180-year-old Pachaiyappa College, the PG Department of Botany has already completed 75 years of excellence and is gloriously marching towards its centenary. Having been aided by rich contributions from scholars like our former Professor V.S. Sundaralingam and many of his successors, the department strives hard to be in the forefront of academics. I'm sure any academic exercise taken up by this department will be of immense importance to all those who undergo it. Today, they have arranged for an international webinar on recent development in genetics and genomic medicine. The learned resource person will make it as an enlightening one, I believe. I congratulate the members of the department, especially the organizing committee, for taking the efforts for this important academic event, which will be more useful to all the participants, I believe. I wish this webinar a grand success. Thank you all.
Thank you, madam. Thank you very
madam.
much for impressive and realistic presidential address. Now I can call upon our organizing secretary, Dr. M. Ilamvazhi, Assistant Professor in Department of Botany, to introduce today's resource person and to enlighten the purpose of webinar. Over to M. Dr. Ilamvazhi. Please, sir.
Thank you, Dr. Kavina. Very good morning to one and all. It gives me immense pleasure to introduce Dr. Rangasamy Erumalai, Senior Application Scientist, Agilent Technologies, California, USA. Dr. Erumalai obtained his PhD degree from Center for Advanced Studies in Botany, University of Madras in the year 1995. He moved to the US for postdoctoral research in 1996 and has worked in the University of North Carolina and the University of Arizona, where he designed the genome-wide long oligo microarrays for maize. Since 2005, he has held various positions in biotechnology industries and is currently in Agilent Technologies. He is an expert in next-generation sequencing and microarray technologies. He has conducted several international workshops and trained numerous researchers on NGS and microarray applications. We now invite Dr. Erumalai to deliver his lecture on recent developments in genetics and genomic medicine. Over to you, sir. We are eagerly waiting to listen to you.
Okay, can I go now? Can I go now?
Yes.
Okay. Thank you. Thank you. Good morning, and thank you, everyone. I would like to thank Principal Madam and also Akila Madam for inviting me and Kavina Madam and Ilamurdi to coordinating this seminar. I really thank from my bottom of heart. And it is a pleasure to be here, and also good to know Akila Madam is my senior from CAC in Botany. It's nice to have some kind of connection over here. Fantastic. So let me share my screen, and then I can go from there. Can you see my first slide?
Yes, sir. Yes.
Okay, well, fantastic. So anyway, thank you very much for giving the opportunity to share some of my experience as a well, biologist in the first, and then metamorphosed into a molecular biologist. And so before jumping into seminar, I would like to give some perspective to the young students. I came from a very small village near Thiruvannamalai. I went to government school and I went to Thiruvannamalai Arts College. Then I had an opportunity to go to do my master's in Annamalai University. Then I had an opportunity to do PhD with Professor Mahadevan. The reason I'm saying this one is for my students, right? Whenever you have opportunity, you have to hold on to, and then you have to work as much as possible, work extremely hard. You'll get to only one opportunity to prove yourself. When you have opportunities given to you, do not let pass. And also do not hesitate to work hard. Like there is nothing called I worked hard enough. There's nothing called like that. You can work. I used to work like 16 to 18 hours when I was doing PhD and postdoc. So there is, you always work hard, learn all the time. Actually, I am a trained botanist. Well, I studied botany from BSc and MSc, and did PhD in plant pathology. But right now, past 10 years, I don't do anything close to botany. So do not think that what you study is the only limitation. You can shine if you have opportunity. You can move into new areas and then learn many things. So today, I would like to give you a little brief overview about recent developments in genetics and genomic medicine. This is a very important topic, I think, because genomics and then medicine has been moving very rapidly as we all going through COVID, and we know how fast the COVID vaccines are developed and the testing is going on. So I will be talking some of those things as well. And so before jumping into detail, I would like to give you a basic definition of what is genetics and also what is genomics, right? The genetics is a branch of biology concerned with the study of genes and genetic variation and heredity in an organism. That is a traditional way of doing things. And what is a genomics? Genomic is interdisciplinary biology focusing on the structure, function, evolution, mapping, and editing of genomes. And you can ask, what is a genome, right? Genome is the genetic fingerprint or genetic material of every living organism, starting from virus all the way to human being. Everyone has a genome. It can be DNA mostly, and it can be RNA, like you know that COVID. And what is a genomics? Study of all the genes in an organism and how the genomics become feasible nowadays. That's the topic of focus today. So here's the small milestone in the genetics and genomics achievements. We all, I think, heard, I'm sure everyone heard about Darwin and origin of species. I don't know how many of you had an opportunity to study, actually read that book, Origin of Species. And I had opportunity to read only when I came here as a postdoc because I didn't have an opportunity to buy the books and all. So one of the fantastic book in biology, every biologist should read it. It is a fantastic book. It reads like a novel and awesome book. That changed the biology forever. And also as a genetist and biologist, we all studied Mendel. Gregory Mendel is the father of genetics, and he's a monk. He was studying the pea plants. Like when he was growing the pea plants, he was observed variation in their characters, in the seed and then flower color and all those things. And then he was curious enough to ask the question why there is a difference and how it happens. So he is the one did the basic crossing between the round seed and the wrinkled seed, and then we studied about 3:1 ratio or 2:2. So 1:2:1 ratio, right? So one of the lessons I always take it from that one is, you always, when you want to study anything or you want to improve something, you look around, find problems, are curious, something it strikes you, try to develop and understand that problem, try to provide a solution. That's the science all about. So there are a lot of problems every day we encounter. And then if you want to understand, if you let your curiosity to understand the problem, that's what bottom line of the science. Anyway, the next milestone is the nucleic acid. In 1871, Friedrich Meiska, actually he was a German scientist. He was isolating some, working on some cells, and then he isolated some new compound, and he didn't know what it is. And then he called it's a nucleon because it came from the nuclei. So it ended up in the nucleic acid or DNA. And then, the famous Avery Oswald from New York, and he is the first person proved that DNA is the genetic material by transforming the virulent factor from pneumococcus you kill, and then mix with a live non-virulent pneumococcal cells, and then non-virulent pneumococcal cells become virulent, and then he proved that the material that transferred to the new bacteria was heat resistant. We all know that protein can be cooked at 80 degree, 100 degree, and it never gets back to normal structure. So even after heating, the genetic material get transferred. So he proved that the genetic material that was in the belief of protein has been disproved, and then he proved that DNA is the genetic material. And we all know Watson Crick, right? 1953. And another great book I recommend everyone to read is a DNA Double Helix by Watson. And he's one of the greatest scientists who ever lived. And 1953, Watson Crick changed the biology forever. Biology was never the same after 1953 because they proposed the structure of the DNA, and the structure is address everything, and then on the genetics and then genomics picked on. And 1961 to 67, the genetic code has been revealed by a group of scientists, including one of our famous Indian scientists, Hargobind Khorana, and he got Nobel Prize for that. And then in 1970s, restriction enzyme was discovered, and that is helped to progress the cloning and molecular biology. And 1977, Frederick Sanger from Oxford University devised or invented the Sanger sequencing. Basically, he's one of the that Sanger sequencing is the founding of all the genomics. Without that, no DNA sequencing could be done, and whatever happened past 50 years, 30, 40 years wouldn't have happened. So he was basically a chemist. He devised the way that how you can able to identify DNA sequence easiest way. So if you take a piece of DNA and if you want to make a copy, all you have to do is add some starting material as a primer. It's called piece of DNA, single-stranded DNA, and then add some nucleotides A, T, G, C, and then the enzyme in the buffer. And the enzyme will copy the new DNA from the old one. That's what called DNA replication. And the DNA replication happens from joining one nucleotide to another nucleotide and another nucleotide from 3' to 5'. That's what the enzyme synthesizes. And if you add dideoxynucleotide to that reaction, the extension of the DNA synthesis stops. That's how you could able to randomly synthesize smaller fragments, and then you can add, if you synthesize a DNA in one reaction, you take all the ATGC and then add some amount of dideoxy A, and then dideoxy, and another reaction dideoxy C, like that. And then if you separate on the gel, you can able to read the sequence. I will show you the next slide. And that's what Sanger sequencing all about. And then 1983, Gary Mullis kind of invented the polymerase chain reaction. And then 2003, Human Genome Project completed. Actually, the Human Genome Project, I'm going to talk about more in the following slides. I don't want to go more. And now, actually, there are a lot of tools available. The latest tool is CRISPR, that is the greatest genome editing tool. It has a lot of applications in the human medicine and crop science and all field of biology. I'll be talking at the end of the talk about that. So I will be, if I'm going too fast, you can let me know. I can slow down. And if you have any questions, please stop me and I can answer you. Okay? Everything is okay? All right, I will move forward. So before going into details about genomics, I also want to give a small piece of information, that is a genome size. What is a genome and what is a genome size? So every living organism has genetic material that's called a genome, and not everyone has same amount of genetic material. For instance, bacteria has only 5 million bases, right? And the yeast, that another single cell organism, that has 12 million bases of genetic material. It may be arranged in 16 chromosomes, but doesn't matter, but the total amount is important. And other examples, I can keep going on all those things, but for us, important thing is some of the model organisms like Arabidopsis, Drosophila, rice and maize, and human barley and wheat. Arabidopsis has 125 million bases, one of the smallest genome. That was the first thing was sequenced before human genome, actually. And then you have Drosophila, 180 million bases, rice, 400 million bases, and the maize, like 2.5 billion bases, like 2.5 gigabases. And the human, we have 3 billion bases, and that's a haploid, and then 6 billion bases as a diploid. And barley has 4.9 billion bases, and wheat has 15 billion bases. Wheat is one of the largest genome, and it is difficult to sequence. Still, the full wheat genome is not sequenced yet. And also, wheat has hexaploid, so the complexity goes more. And another tidbit of information about genome size, you have seen that even within grasses like rice, barley, wheat, and maize, you have so much variation in the genome. So why there is a huge amount of variation? And you can also see that Arabidopsis has the smallest genome, like 125 million bases. What causes the increase in the size? So typically, transposons, like Barbara McClintock studied about the jumping genes, right? That's called transposon. They randomly go and insert. When it inserts, it doesn't look for no preference. So it can mutate a gene. So when a gene, important gene, gets mutated, the plant may die. So to avoid that one, basically the plant does is homologous recombination or segmental duplication. Basically, it just duplicates its original gene and then having the mutated copy like that. That's how the genome size get increased. Like maize has a huge genome actually. And also the ploidy. You have diploidy is a common, but plants have a lot of polyploidy, like banana and then wheat. They have a lot of polyploidies. And also the other way around, the genome size can be smaller. How it happens. So that is a very special character, that is organism can lose its genome when try to evolve in a parasitic environment, because we all know that parasites do not have to run and fend for their food, and they don't have to photosynthesize, they don't have to absorb water, nutrition, nothing. So it basically takes the nutrition from its host and lives. So they don't have to have large genome, and that's why they kind of lose their genome size. One of the actually interesting examples are the chloroplast and mitochondria. Chloroplast has its own genome, mitochondria has its own genome. Chloroplast has only 110 genes, whereas mitochondria has only like 37 genes. You can think about it. One time, millions of years back, even longer than, they might have been a free living organism, either a bacteria or algae or something. Like when they incorporated into higher organism, they're slowly losing their genome. And also, you know that like a viruses has a very small genome, and the mycoplasma also smaller genome. All right. So this is just a brief overview of genome sizes, why that matters, and how the evolution happens, and then what is the pressure. All right. So everybody clear so far?
Yes,
can you hear me?
Yes, sir. Yes. Questions will be taken at the end, sir.
All right. Okay. Just positively respond somebody, because I don't know whether I'm connecting or not. Right?
Yeah. Sure.
Okay. All right. Cool. And this is the Sanger sequencing. I was explaining Fred Sanger invented the sequencing. So this is very, very important, because as you can see, Sanger sequencing, you can make a DNA replication. When the replication happens, you can add dideoxynucleotide in the reaction. Like you typically, I used to do it when I was in postdoc in CCMB. It takes about like 3 days to get a 300 bases of reading nucleotide. So basically what you do, set up 4 different DNA synthesis reactions, one with the dideoxy A, dideoxy SIA, T, G, and C. And then what you do is you separate them on long polyacrylamide gel. And the primers that we use to make the new DNA labeled with radioactive P32. And after electrophoresis for 18 hours or 24 hours, and then you transfer the gel and expose to X-ray film, and then you, after 2 days exposure, you develop and then read it. Basically, you can read it here, the base A, T, A, and there's a C, T, C, A, like that. One person will be reading, another person will be writing. Somebody makes a mistake and you start over from the beginning. So it's a fun thing, but it is very tedious, very expensive. And from that gel electrophoresis, ABI come up with the capillary electrophoresis. And they also, instead of making 4 different reactions, they came up with dye incorporation reactions, like different colors. They can be excited with the different lasers, so red, green, yellow, and blue. So when the dideoxy chemistry moved to big dye chemistry, you can able to separate them in the capillary longer, and you can able to read up to 1,500 bases, right? So that's what ABI came up with. Let me get the pointer options. This good? Okay, yeah. So they come up with ABI that can give 500 kilobases of sequence in a day, and then they came up with the ABI 3730 that is 96 capillary. It can give about 2.8 megabases per day, because you can run about 3 or 4 reactions. So that is how the Human Genome Project started. So in 1990, when I was doing a graduate student in Chennai, and we saw the announcement Nature, we got like 3 months later the Nature journal, right? We all excited, and then we didn't even know what is the Human Genome Project, and we all read, and then we are so excited, and then want, wow, that is great, because Human Genome Project was is a greatest undertaking in biology to decode the entire genome base by base, right? It was initiated by US government, and then it was completed in 2003 after 13 years. And it was a multinational collaboration, US, United Kingdom, Japan, France, Germany, and China. And there are more than 20 labs running this project. The major effort was coordinated by NIH. This project costed about $2.7 billion. One of the largest biological project ever run by US government. Biology programs never go billion. They will be like millions or hundreds of thousands. But first ever billion-dollar project was the Human Genome Project. And when the sequence, before the sequencing was complete, there are a lot of predictions that, oh, there will be 100,000 genes, there will be 50,000 genes, or there will be 200,000 genes. To everybody's surprise, when they complete the sequence, when they did the annotation, that is the predicting genes, it was much smaller, like a 25,000 genes. I still remember it was like a 25,000 gene, and then it defined to 22 to 23,000 genes now. So that was one of the greatest achievement by US government, or even the human being decoding the Human Genome Project. And during the Human Genome Project, there was a 10-year project and multiple labs. It was a systematic approach. That is, you have a human genome, there are 23 chromosomes, and then you make the libraries for each chromosome, and then try to, not every chromosome, they used to make yeast artificial chromosome libraries that will be 400 kbs kind of stuff. They try to map them on the chromosome, and then make sequencing for individual yeast libraries, and then you align them, because you can align them easily, so that when you align those yeast artificial chromosome libraries, and then you make the alignment, you can easily stick them together, make a long reads. So this is what took a really long time, because it's a systematic approach. During that time, there is a guy named Craig Venter. He is a kind of out thinker. Like he always said, there's a statement called "always think outside the box," right? So he always outside the box thinker. So instead of going through this step-by-step process, he said, "Don't worry about it. Let's sequence everything. That use the computer to align the small piece of DNA." So what he did was that he started a company called Cellular Genomics. He basically completed the entire Human Genome Project in 2 years, when the 6 nations, like 20 labs, took 13 years. So his approach, that is, it's called shotgun sequencing. That is, you basically take the genome and then chop into small pieces and then sequence them. Once sequencing is done, let the computer align the sequences and then map it, and then we can able to piece them together and get a big picture. So that is the greatest thinking, and that changed the genomics forever, because based on his idea, he proved that idea worked, and then he sequenced Watson genome. He got the James Watson. He still lives in Long Island. So he got his genome, and he sequenced his genome, because he's the one built the DNA structure. And he did it within 2 years. And this idea of capturing small sequence, and then you can able to pile up to make a big thing, is the foundation for next generation sequencing that I'll be talking a little bit more later. So after completion of Human Genome Project, so we well and good, we finished the human genome, and what is the next? That's the question they were asking about. So the reason why the Human Genome Project was initiated was, if you sequence the entire human genome, you will understand. You will understand all the genes. You will understand all the genetic diseases that a lot of people you born with, and what causes the genetic diseases, and how to address, and how to find out, how to cure, how to develop drugs for those things. So that is the obvious next step. That is, identify different group ethnicity in the world. Like there are a lot, hundreds of ethnicity, and each groups have their own unique character. Some people are resistant to malaria. Some group ethnicity, the ethnic people are very sensitive for smallpox, and Asians are sensitive for alcohol. So there are so many different genetic variations. So it's called HAP map project. That is, the human HAP map project is identify different ethnic groups and then try to sequence their genome and then try to find out the genetic makeup of different evolutionary significance of these ethnic groups. So this project was started in 2002, and then it's kind of done in 2010. But this project actually started with a small, and they end up in 1,000 genome project. That is, instead of sequencing few important people like James Watson or Greg Venter, they want to sequence minimum 1,000 people's complete genome, and they are representing different ethnic group. For instance, this 1,000 genome project also included 2 ethnic group from India, and there is one ethnic group from Sri Lanka, and then one from Burma. And you can see these are different ethnic groups. They selected not one individual kind of family, like you can see their numbers, hundreds of people from each ethnic group. They try to identify some people with the genetic disorders, some pedigrees with some resistant to genes. So they sequence those things, and then they try to put understanding of disease causing or resistant phenotypes for different ethnic groups. So this project almost done actually. This is one of the important project that after Human Genome Project was done. So because of the Human Genome Project, there are a lot of things happen. Like example, HapMap project, 1,000 Genome Project. But I want to give you 10 highlights, that feature points of the Human Genome Project. So these are 10 breakthroughs came from Human Genome Project. Because Human Genome Project is such a large genome, that basically helped to drive sequence of other genomes. So like almost a lot of plants, a lot of animals, like bovine, dog, cat, and mouse, obviously mouse, rat. All those genomes have been sequenced. Like now genome sequencing has become very easy, very simple, actually simplest thing. And then all the data is publicly available because it's so, all the tools are there, and then you spend a little bit money, you can utilize the tools, and then you can publish that, and then all the genome sequence are available. And then evolutionary significance of human race, and then newer, faster, and cheaper way of sequencing genes. I will be talking about that. And then finding the causes of diseases. We all have a lot of, I'm sure that many of you have relatives, and then we encounter or heard about genetic disorders. And we all know the problem of cancer, right? So all those things, like finding cause of diseases, and also life-saving genetic test, and also diagnosis, and that can lead to effective medicine and more personalized drug treatment. I'm not going to talk too much about that because it's there's time limit, and because of the Human Genome Project, the genome sequence available, and we can able to sequence individual genome or exomes, like only the coding sequences, identify your genetic makeup, and then you can plan in case if you encounter some kind of difficult illness, we can define or devise proper treatment. And also, genome mapping is a kind of bargain. Like you can actually, when I was doing a PhD or even the postdoc, I used to pay $1 per base to sequence. Now, I'll be having a thing. This sequencing cost so cheap, and you can sequence your own just by simple as, if you're curious, having $1,000, you can sequence your own genome without any problem. And then now the latest is the gene therapy or genetic engineering. I'll be talking about that. So I'll be talking more detail about the ways to sequence the genes, finding the cause, or and also disease diagnosis. And at the end, I will talk about the gene therapy. All right. So far, so good.
Yes, excellent, Emily.
Oh, thank you, Murthy. I didn't know that you are here too.
Here, I requested the organizer to unmute my mic, so they did. Thank you. And yeah, excellent. They were afraid of the background noises. That's why they unmuted all the participants.
That's good.
Yeah.
Thank you. So far, so good, right?
Yeah.
All right, cool. So new, faster, and cheaper way of sequencing the genes, or DNA sequencing. This is the slide I used to present for a lot of people. And this is kind of timeline. Okay, now it's fine. A timeline of next generation sequencing. So when human genome sequencing done, the Human Genome Project was exclusively done using Sanger sequencing, that is ABI 3700. And I used to have a picture that Cellular Genomics used to have rows and rows of 3700 sequencer, and it used to be like a factory, like running 24/7, and they used to make pumping out the sequence. But still, the throughput was so small. And after that, and also like Craig Venter's thinking about that shotgun sequencing, basically changed the world. That first Roche 4x4 came up with the idea that instead of sequencing one piece of DNA, try to sequence multiple pieces, like hundreds of thousands of pieces of DNA at the same time. So 4x4 came up with the idea that they used to have a beads, and then each bead will coated with the one piece of DNA, and then like that you can have 10,000 beads, and then you sequence all 10,000 pieces at the same time, adding one base at a time. And so now instead of one piece of DNA you're sequencing, you have 10,000 pieces of DNA you're sequencing. So that much throughput increases. That's why it's called next generation sequencing. And then in 2006, Genome Analyzer, or Solexa Genomics, they come up with a different idea. And then that company was bought by Illumina. They're the one developing major player sequencing platform right now. And then ABI came with solid sequencing, and this solid sequencer is completely disappeared because they could not able to survive. And Illumina came up with the HiSeq in 2010. Now they come up with NextSeq. This can produce like a HiSeq in 2010 that can produce about 750 megabase, 750 billion bases of reads in 10 days. If you think about Human Genome Project, it took 13 years to produce 3 billion bases, like 3 billion times 10 times. It took 10 years, whereas this guy can produce 750 billion bases in 10 days. And another technology came, Ion Torrent. This technology came, instead of doing sequence by synthesis, like adding one by one, they used to test pH change in microvalves. They can able to identify what kind of base is added like that. And this Ion Torrent is a very revolutionary idea, but this is not a major player in the market right now. And most importantly, we have to focus on Illumina, and they came up with the X10 NextSeq, and now they have newer sequencer called NovaSeq. This is the latest sequencer from Illumina. This machine can produce 6 terabase of sequence in 2 days. I just want to give you some perspective. That is 1,000 bases called kilobase, and then a million base is called megabase, a billion base called gigabase, and then billion, what is the next level? Trillion bases. So trillion bases called terabase. So this machine can produce 6 trillion bases in 2 days. Think about the DNA sequencing came from the days when I was doing it in 1995, took about 5 days to produce 350 bases. Now I can produce, I can produce 6 terabase sequence in 2 days. So that is how the DNA sequencing has metamorphed, and because this enable a lot of new things, and Illumina knows that we can sequence individual genome with $600, basically $600. All you need, $600 in American thing is not a big deal, because if you go and buy an iPhone, it costs $900. So by spending $600, you can sequence your genome and identify is there any risk you have or something like that. Actually, I will talk a little bit more common practice now in here about human health, and also like having babies and all. So the sequencing has evolutionized very rapidly, and then 2 other sequencers right now are available. One is PacBio. They could able to sequence very long sequences. This machine can sequence only like a 200 base into 2, like a 500 base in piece of DNA. But a PacBio can sequence 10,000 bases. That helpful to align them new organisms, and also like a telomeric region and centromeric region, because those are repeated regions. So here, another graph I wanted to share with you, that is the cost of sequencing, actually. This is from 2001, about $10 million for megabase of sequencing, that is by a conventional sequencer. Now, here is 10 cents per megabase. That is 0.01 cents per base. So it's kind of the technology is moving so fast, evolving so fast, and also the cost of the sequencing coming rapidly low. And now you can everyone able to sequence their genome and understand if you have a genetic risk in your family or something, you can able to manage your life better. So this one, I'm personally involved, because this technology was developed by Agilent Technologies. When the sequencing cost was very high, that is around 2007 and 8, Agilent Technology, we used to make a lot of microarray, and we use inkjet printer to synthesize DNA. Yes, what I said is true. We use the inkjet printer that you print a piece of paper, right? We use similar technology to make DNA. We can able to make 500 base long piece of DNA with inkjet printer technology. So what Agilent came up is we had a collaboration with Broad Institute in MIT. I think Dr. Murthy was working Harvard as a neighbor. Because the whole genome sequencing still was expensive, and if you sequence whole genome, you had to sequence at least like 30 to 50 times, and you produce so much data, and then handling those data, aligning, making sense is difficult. So if you could able to capture only the coding sequences, like if you take the human genome and the 3 billion bases, and if you look into the human genome, only 50 megabase, right? 50 megabase, like 2% of the genome is responsible for all the protein coding. So if you can able to just take only those things and then sequence them, then you have all the information to understand about genetic makeup of any individual. So that's what we came up with. It's called Sure Select technology. That is called otherwise targeted sequencing. So the way it works is you take your genomic DNA and then fragment them, chop them to small pieces, and then make a library, like add some sequences to amplification, and you make a single-stranded DNA using our inkjet print technology, add biotin in that. Now you mix this baits, we call it baits, mix this and then library together, and then heat denature and let them hybridize for 90 minutes or 16 hours. Then mix with streptavidin coated magnetic beads, and then streptavidin binds to biotin, and then you can use the small magnet to pull down. So because we put only coding sequence, exonic sequences here, so this will bind only exonic regions of your genome. So other 98% of genome, we don't care at this point, because if you want to understand any mutation in the genome, you have to sequence only the exome. So when you pull down, I'm pulling down only the exonic regions of the human genome. So after pulling down, wash it out, all the unbound things, and then amplify and then get it sequenced. So this process speeds up the sequencing because it's very small, and also the handling the data, sequence data, because you still need to have supercomputers or really powerful computer to align in the anti-human genome, right? So any simple computer can able to align that and then give you clear idea about what happens. And also you can able to sequence much deeper, like you can sequence 100 times or 1,000 times or even 10,000 times. For instance, if you want to understand the cancer doesn't come from the mom to kid, it can happen somatically. So if you identify the cancer causing mutation early in the cell population, then you can able to devise a cure. So this helps, this select technology helps to sequence much deeper and able to identify mutations even the lowest frequencies. And when that is the technology enabled to identify a lot of mutation. I will talk about that one a little later, next chapter. So now the sequencing technologies, like you have whole genome sequencing, 1000 genome project, and SureSelect technology, you can able to sequence them with very, very minimal effort. But what are the questions you can answer? Still, there is a lot of questions you can able to answer, is because if you take if you want to understand tumor, so if you get the small tissue from biopsy sample, you sequence it, and it gives you overall population, but it does not give me what are the population of different types of cells, like I say, heterogeneity of your tumor, or do you have the T cells? How many T cells are there? How many memory B cells are there? How many types of mutations are there? So it may give you overall picture. It's not going to give you exact number of picture, right? So the sequencing technology become much more sensitive. Now you can able to sequence one single cell. So if you take a piece of biopsy sample, and you can able to identify sequencing information of entire like a 10,000, 100,000 cells in the tissue, and individual cells, what information is. It's called single cell sequencing. So basically what you do is you have a bead having attached to some kind of barcode in it, an oligo, and you mix that bead with a single cell. You basically release the cell from tissue. You can encapsulate that bead and the cell in a small lipid capsule, and you run through a library preparation. Either you can prepare the DNA library or RNA library. And after RNA library is prepared like this, and you can release this lipid capsule and get all the DNA and sequence, because each bead has a barcode. All the DNA pieces coming from that particular cell will have one barcode. That is, barcode is, yes, 10 base of ATGC kind of sequence. Like that, different beads will have different barcodes. So all the piece of DNA coming from one cell have same barcode. Same way, you can identify hundreds of thousands of cells, and then you can able to estimate if you get the sequence from one cell, and you can able to figure out what type of cell it is. Is it a memory B cell or T cell, or there is a one type of clone of cancer or a second type of clone of cancer, how many things like that. So now the technology is so powerful, you can able to do single cell sequencing. So that is latest one going on. This has been going on about like 4 or 5 years, that is single cell sequencing. And even better, this is the latest one. It is in like a couple of years right now. It's a spatial transcriptomics. So what is a spatial transcriptomics? What is the difference between single cell and spatial transcriptomics is, what you do is you take your biopsy tissue material and put on a glass slide. The glass slide has bunch of spots. Each spot has a barcode, and you put your piece of tissue and then stain it with whatever the stain, and then take an image. And then after taking the image, what you do is you basically release the break the cells down, and all the RNA will be released. The RNA will binding directly below the barcode, and you can make a library out of that. And then after making the library, you can pull this DNA and then sequence it. After sequencing, because each cell gets its own barcode, you can able to tell each cell what kind of cell is, where the cell is, and where the new clone is forming in the cancer, and where the B cells are accumulating in the tissue, and where the T cells are accumulating. So that kind of image, actually, that there is a secondary level of image, like one, you have a physical image of cancer morphology, and then you can superimpose that one with genetic image or genetic typing of each cell, and then it gives you much more clear picture, and then you can able to handle the treatment regime of the cancer treatment. All right, so far so good?
Yes, indeed.
All right, thank you. All right, let me go forward. And next chapter I want to talk about is finding the causes of disease. Am I running slow, or is it okay, Face?
Okay.
Okay, sir.
Okay, cool. So finding the cause of diseases, right? So traditionally, the way you find a disease, one of the example was cystic fibrosis. It took about 10 or 15 years to identify the disease causing mutation for the cystic fibrosis. But now, oh, there you go. There are well over 6,000 genetic disorders are cataloged right now. And there's more genetic disorders are constantly described and also cataloged. And out of the 6,000 disorders, there are more than 600 of them can be treatable by drugs. And in 1 in 50 people are affected by known single gene disorders. So you can read those things, okay? So there are a lot of problems in the human population, right? Almost every other person has some kind of problem. And how do you find out? And one of the thing is either is that the problem comes from genetics, like it came from your DNA, is to sequence and identify. So when we released our exome sequencing, that's your select exome capture sequencing, and there are a lot of publications came immediately to identifying the causative mutations. And every month there will be like 3 or 4 publications will be coming. And it was kind of a very exciting time because, as I said, for cystic fibrosis, it took 10 years, several labs to identify one mutation. Now, with the power of DNA sequencing, as well as power of Sure Select, you can able to easily identify causative mutations. And here's the simple example that in the 10 years when we released the Sure Select product in 2009, from 2009 to 2011, there are like 50 genetic mutations have been identified related to different syndromes. So it was one of the greatest result of Human Genome Project and the DNA sequencing technology. Now we know what is a mutation is. So we can able to target those genes, identify that, and then you can easily manage your lifestyle or devise a cure. Or you can also, if you don't have a drug, you can use this information to build in a pathway and then find the cure or find the drug that could help to address that particular problem. So life-saving genetics test and more effective medicine, right? That's my next topic. So genetic diagnosis, disease diagnosis is most important. If you don't diagnose your disease properly, you're never going to get the right medicine. If you are diagnosing a viral a disease to bacterial, then you'll keep pumping the antibiotic, never going to cure. And if it's a bacteria, and then if you think it's going to be cured itself, you're never going to cure, you're going to kill. So there are a lot of things. And also the genetic disorders that originate from the mutations. And if you understand what mutation is causing that problem, and you can able to find out a right solution. So nowadays, exome sequencing becoming routine for disease application. Actually, I have next slide to show you about that. And also targeted panels are becoming common because nowadays it is acceptable in many insurance companies that if you have insurance, if you have some kind of symptom, the doctor can prescribe to sequence that small panel and they get reimbursed. So it has become common practice nowadays in healthcare industry. And also for the cancer, there are 550 genes are involved that has been well defined. Actually, more mutations are coming, and there are panels or cancer panels are available, can be able to sequence. And actually, cancer diagnostic have been taken much more rapid leap in the technology. I will be talking a little bit more later. So this 550 gene cancer screening panel can identify point mutations, deletions, duplications, translocations, gene fusions, alternate splice variants, and also microsatellite instability and tumor mutation burden. So these all type of changes can be identified using these small panels. For instance, translocation is one of the important cause for certain kind of cancer, and also gene fusion. And another important thing nowadays is recommended by doctors, oncologists, is microsatellite instability. So what microsatellite instability is, when cancerous mutation happens, it changes a lot of things, and it rapidly growing, and it keeps multiplying the genes. And during that one, there will be a lot of errors happen. And typically, if you think about it, our cell cycle, you have one cell to second replicating two cells, the genome is replicated. During the replication, you will introduce errors, and those errors are rectified by mismatch repair system, right? So if without the mismatch repair system, you will not have identical sequence in all over body, and you will have a lot of mutations. That's what happens in the cancer cell. So one of the easiest way to find out is microsatellites. Microsatellites are small repeats close to the telomeric region and the centromeric regions. And there are like a 250 or 300 sites that you can able to identify. And because of the mismatch repair system is compromised in the cancer mutation, the microsatellite will be unstable. You will not have proper copy. So if you identify that microsatellite instability, you can able to devise your treatment. One of the findings is like if you have high levels of microsatellite instability, nowadays oncologists recommend for PD-L1, like what is that? PD-L1 blockers, immunoblocker therapy, right? So These high stability cancers respond much more for immunoblockers. The same like a tumor mutation burden, that is how many tumor mutation in 1,000 bases or 10,000 bases, that helps to devise the proper treatment for the cancer. So there are several panels, and also at this point there are more than 2,000 genetic tests that people can take to understand the risk, right? So this is actually happening very rapidly, this new finding from 2020, that newborn screening, right? So when my daughter was born in 2000 in Arizona, they took some blood sample from her. And the reason was, in America, the state government health system, they screen for some of the basic disorders like polyketonuria, and then maple syrup syndrome, some other things, by looking in the chemical signature in the blood, right? But those screening is good. I'm not saying it's bad, but those are targeted for handful of disorders. But now you have the capacity to sequence the entire exome, human exome, with affordable thing, and that can provide you all the mutation that happen that is not able to do understand by blood test. So they actually done in for newborn babies, and this exome sequencing going to be recommended in the future, I'm sure, within like a 3 or 4 years or maybe 5 years, that exome sequencing for newborn babies is going to be routine thing. I know for sure in Australia, and one of the states Agilent is working, they already implemented that exome sequencing for newborn babies, and because they can able to identify errors in the metabolism in the system, right? So this is the greatest advantage that human genome project and next gen sequencing provided to humankind. So think about it. In 5 years, you can you're doing routine test like that. You can also do your own routine genome sequencing. Find out what's wrong with you, right? So other one, actually, this is not new means like this been like about like 7-8 years, liquid biopsy. So what is a liquid biopsy is, so biopsy, we all know that, right? If you go to the doctor and then you see there's a small bump in your face or some kind of growth, and if you feel like, oh yeah, maybe tumor, and the doctor say, okay, let me take a biopsy, right? So he will take a pinch of tissue and then he send it for testing. So instead of doing that, you can do very similar kind of screening by drawing few milliliter of blood from your body. That is, what that means is, when the cells in your body die, they release their DNA into the blood. And not only normal cells, if there is a tumor cell, they die, they release their tumor DNA in your blood. And same way, if the mother is pregnant, the baby's DNA gets released in the mother's bloodstream. So there's a lot of potential. Like here, you can see here, tumor cell, non-tumor cells, non-tumor DNA, tumor DNA, RNA, and also extracellular vesicles for chemicals. So if you draw a little amount of blood, and you can identify all these things. So this is actually becoming very, very, very common practice nowadays in Western countries. Actually, I'm sure India also has become common now. It's something called non-invasive prenatal testing, otherwise called NIPT. So what is the NIPT is, they basically take about 3 milliliter of blood from mother, and then as soon as you draw the blood, they spin down, remove all the white blood cells, and then they'll take only the plasma, right? Because plasma is the one without any cells. And they isolate the DNA from the plasma, and then they sequence the DNA. right? When you sequence the DNA, you can able to identify chromosome copy numbers, because if a normal chromosome, you have to have 2 copies, right? So when they sequence, they look for aneuploidies, that extra chromosomes, because the mother blood also contains the baby's DNA. If there is anomaly in the baby, that's going to be shown in your mother's blood. So basically, they look for aneuploidy, like for Down syndrome. We all know Down syndrome. There is extra 21 chromosome, and then trisomy 18. Trisomy 18. If you have trisomy 18, baby won't born alive. And also trisomy 13, and then extra X chromosome or Y chromosome abnormalities. And also they also do targeted genes, like Natera. There's a company, actually it's about like 10 miles from down our home. They do very this kind of prenatal testing. And I know there's a lot of women who go through the pregnancy. They take this test because it doesn't cost a lot. It's less than $1,000 per test. And you have peace of mind because you know your baby is not defective or anything. So, or if you know that your family has some kind of genetic problem, and I would certainly advise to take it, because you will make sure that baby is not going to be suffering the rest of their life. And actually, it's not even worry about your family has a problem. It's better to take it. It's less than $1,000, less than your cell phone. So you're going to bring a human being, well, and healthy human being. So I would certainly recommend anyone going to have a baby will take this kind of prenatal testing, and that way you have peace of mind for the rest of your life. Everybody's good?
Yes, sir.
Yes, sir. Yes, sir. Yes, sir.
Am I making you sleep?
Yes, sir.
Yes?
No.
No, sir.
All right, okay. Sorry, I sometimes carried away. I just keep talking because these are my interesting subjects. All right. So I will go to the next slide. This is one of the another most important thing I want to talk to you about is non-invasive cancer screening. So same like I told you here, that circulating tumor DNA. So if you draw a couple of milliliters of your blood and isolate the DNA and you sequence them, you could able to identify, is there any mutations in any of those cancer causing genes? Because the cancer cells also die. Even though they grow rapidly, they die. When they die, they release the DNA into the blood. So if you could able to pick out any of this mutation as early as possible, you can take action. That is, you can take action of go on full screen, or you can immediately see the oncologist and then devise a regimen of treatment, because early detection is always better. So there are, I'm giving a very few examples. I'm giving only the America, not even full list. There's a very partial list, just for examples. There are multiple companies. Actually, in India, there is a genomics company. I work with them. Actually, they have a branch in here in close to Bay Area. They do a lot of exome sequencing, targeted sequencing. So anyway, Foundation Medicine as one of the FDA-approved liquid diagnostic test that targets about 300 cancer-causing genes, and also Guardant Health 360. They target 74 genes, and there's a new one, Lunar 2, that has about close to 500 genes. And then Grail, this is a company funded by Illumina. They have a different technology. They detect methylation. They could able to identify 50 different how to, oh man, I've been keeping this one open. Sorry, I don't know if this one going to be there when you're recording. Sorry for that. There are about 50 types of cancer can be identified early in the oncogenesis. So these companies doing great service for humanity, because and cancer, one of the leading cause of death in the world, and it's a tragedy. And you have to be, whatever you can able to develop test or cure, and that always do that. And also another thing I forgot to add the slide is breast cancer screen. That become very, very, very standard in America, and also like almost all over the world. That is, you screen for only 3 genes, like a BRCA1, BRCA2, and one more gene, and you can identify whether did you inherit any mutation that may end up in having breast cancer. And if you have likelihood of developing breast cancer, it may be better off you remove your breast, mastectomy or something like that. Your life is much more important than just having whatever things, so that is these genetic tests really life-saving, and these things came from Human Genome Project and from using the next gen sequencing basically enable it, right? So the future of cancer management is, like you can see this one. You can have this is my it's not too far actually. In 5 years, maybe even shorter, we go for annual checkup. Right now, if you go annual checkup, they take a blood and then do lipid, cholesterol, and glucose. And next one, they're going to have the exome cancer screening. So every year you go there, your blood you'll be screened for cancer, because this test is much more versatile, because it is not one particular place. It can screen for brain tumor, lung tumors, colon tumors, and ovarian cancer, and skin cancer, bone marrow cancer, everything. So that can be easily identified, and then you can able to manage your treatment regime. So liquid biopsy is becoming one of the biggest tool and fantastic tool for human. So this is like I don't need to go detail. It's like how you do the genotype and phenotype and devise the thing. It's just a slide. Okay. So next and last topic I want to talk to you is gene therapy. And actually, last time when I was in Chennai, Dr. Borang Muti asked me to give a small seminar in IBMS, and I gave the seminar there. It is important to understand what is a new technology is called CRISPR-Cas technology. The CRISPR means clustered, regularly interspersed short palindromic repeats, CRISPR, right? So this is becoming very, very, very important technology. Actually, the inventor, Doudna, got Nobel Prize for this. So there she is, Dr. Doudna. Actually, we collaborated with her in Berkeley. CRISPR, a little bit smaller history. Actually, Doudna was not the one first did it. Actually, it was identified in France in a cheese-making company. That is, certain bacteria able to resist bacteriophage attack, but other batches died because of bacteriophage. And then, when they sequenced, they identified some kind of weird small repeats in the genome, and that time they didn't understand anything. And that kind of evolved. And then Jennifer's lab in Berkeley and her collaborator Emmanuel, she was a postdoc that time, I think. They identified those sequence nothing but immune response. Like actually, if you look back, the restriction enzyme is type of immune system for bacteria. Same thing. This CRISPR is another type of immune system that basically bacteria is attacked by virus. The bacteria, if it survives, it basically takes a small piece of DNA, incorporates in its genome, and then next time somebody attacks, and then it quickly transcribes that region, and then use that transcribed piece of RNA to attack the incoming virus, so that you can kill the virus. So Jennifer's lab basically showed that using this CRISPR, they could be able to cut the DNA into pieces. So what is CRISPR, right? Basically, CRISPR is a small RNA is called guide RNA. So here's a guide RNA and the matching sequence. So this sequence is the memory from viral attack. That this can be engineered to have any sequence in human, any sequence in plant, any sequence in fungus. So this can be replaced, and then you can exploit the system to load it, the target region and the guide RNA, and you have a case9 protein that basically introduce a double stranded break. So the double stranded break is introduced in the virus. If the double stranded break into the virus, the viral genome is cut into pieces and it's not going to replicate. But we can use this technology to exploit, and then you can cut any part of the gene or any part of the exon or any part of the genome. And when it cut, typically the cell responds to repair it. Where how they repair is it uses the homologous recombination method. So when the repair mechanism happens, what happens is if you put a small piece of DNA, that is, if you want to repair a gene, if you put the right copy of the gene, the homologous recombination will basically, this is a homologous recombination donor, it will incorporate and then it's going to fix your DNA, right? So that very simple, actually, very clever idea. That is, you have case9 guide RNA. The guide RNAs will be target sequence. And then if you put that one in the guide RNA and then target sequence and then case9 protein, it'll cut it. And then you also put the proper sequence that you want to repair, and that will be incorporated into a piece of your DNA using the recombination repair mechanism. So now you basically repair the gene. One of the applications now, actually, it has been approved by FDA, is sickle cell anemia. So I'm sure we all studied sickle cell anemia. That is, malarial parasites infects you, and it basically parasitic thing, and you all get fever, right? In Africa, there was some people are resistant to malarial infection, but they are lethargic. They can't walk fast, and they are slow, and their metabolism is slow, because reason is their blood cells become oddly shaped, sickle-shaped cells. This mutation happens in one base change here. That is, T become A, that glutamine become valine. This mutation causes this sickle cell. So FDA approved this one. Is basically you can harvest the stem cell and then isolate the DNA and then repair it and then put it back, and it basically cured the sickle cell. And people have been getting this, and first FDA approved genetic cure by CRISPR. So there are a lot of crazy things going on with the CRISPR. Also, there was a Chinese guy tried to introduce HIV resistant gene for the thing. Those are things going to be problematic, but there are a real lot of cure for CRISPR. And another thing, actually, FDA, without even FDA approval, they could able to genetically modify the button mushroom to knock out the polyphenol oxidase. We all know polyphenol oxidase, right? When you bite apple or other things. One of the best examples is eggplant. If you don't put in water, it will become brown. You know why? Why? Because of polyphenol oxidase. The polyphenol oxidase works quickly and make it brown. So if you knock out one of the polyphenol oxidase, it won't become brown. It will stay healthy longer. So the storage can be longer. And so actually, FDA has refused to regulate it because it's not foreign DNA kind of stuff or whatever. So just putting this thing, there is an application already in the food system. So CRISPR becoming very widely used for curing diseases, and also actually this guy lab, this guy's lab, Zhang's lab, they already developed COVID testing using CRISPR technology that CRISPR can test better than real-time PCR, much faster, less than 20 minutes. And so CRISPR may be the future of disease diagnostics as well. So, that's all I have. Any questions?
Thank you, sir.
You're
It
welcome.
was, hello?
I can hear you.
Okay. Thank you, sir. It was indeed a great presentation. You have given an excellent coverage on recent developments in genetics and genomic medicine. It has sparked a true knowledge on the topic, and I'm sure everyone feels the same. Now the forum is open for the audience to clarify your queries. Please post your questions in the chat box.
Yeah, if you have any questions, please feel free. You can unmute people. And if you have any questions, feel free to ask me if I can able to answer the question.
Hello, myself, Professor Bhuvarahamurthy, HOD Medical Biochemistry. I really thank the principal and the Botany department, particularly Dr. Ilamalidi, for inviting Rangasamy Elumalai to present the recent trend on genomic medicine. Professor, Dr. Elumalai, it's very well done. You are starting from mental theory, Krieger mental. Till the CRISPR technology, it's eye-opening for the students community. I really thank for your presentation. You touched upon the latest COVID technology using CRISPR-Cas. Indeed, this is the needed update or translation medicine, because the RT-PCR and all these things, don't know what's going on. I think this will help. I really thank you for your presentation.
Thank you, Dr. Murthy. Actually, when Ilamardi asked me, and if I can take a talk about individual subject for a long time, but first I want to give you an overview and also make awareness, like actually a lot of things available. That is, cancer screening and NIPT for newborn babies, and the like COVID testing or CRISPR. So these are the new things, right? So it is important everyone understand. I'm sure this is a botany department, but
no, the students from our department also joined, particularly the array used for genetic screening of metabolic disorder or genetic disorder will help them to understand about how this next generation the diagnostic industry is going to work.
Yes.
Great. Yeah.
Yes.
Sir? What, sir? Hello, sir?
Yes, ma'am.
Yes. What is the difference in the efficiency of using CRISPR and RT-PCR? RT-PCR, sorry.
Efficiency, actually, I saw some data that, in any test, real-time PCR is very efficient because you can go few molecules in a sample, right? And they showed that COVID, like a CRISPR technology, can outperform even 1 or 2 molecules in the sample could able to identify.
Oh, that's great.
Okay, sir.
Sir, I have a question.
Yes.
Sir, what is there any relation between this mRNA vaccines and the CRISPR technology? Because
mRNA what? Sorry?
mRNA vaccines based on mRNA technology.
Oh, no. mRNA vaccines are actually, so if you think about the vaccines, right? You basically have to take the protein, and then you have to develop antibodies, and then That's what the vaccines are, right? Even if you look into, you inject that vaccine in your human body, human B cells will remember that, oh, there's a protein, and keep that protein signature as a memory, right? So instead of injecting the protein from external, the mRNA, basically, you inject just mRNA. It's not a gene. It's just mRNA. RNA are much more unstable. RNA can be degraded very quickly. So if you inject the mRNA into a cell, they will translate into protein, right? That protein can be recognized by B cells, and then that B cell will be kept in the memory. That's why the immunity develops. And that's what the Pfizer vaccine is about. And you can also introduce the thing by viral. The viral will not be incorporated into the human genome. So that's what it is. The CRISPR is different.
Thank you.
Somebody is doing something on the computer.
Tanika Salam sir, please mute your mic, sir. Tanika Salam sir.
Sir.
Please mute your mic, sir.
So yeah, CRISPR technology, actually the way it works is, so when the guide RNA binds to the match, it introduces the double-stranded break. That activates the Cas9 protein, and then Cas9 protein randomly cleaves other DNA floating around. So that's why if you have 1 or 2 molecules in the sample, that is enough to identify. That's why it's a little bit more sensitive than real-time PCR.
Hello. Sir, next question. What is the use of GLC in tests, sir?
GLC?
In test. Dr. Arunachalam Pachaiyappa is asking this question.
What is a GLC? I don't know what the GLC is.
What is the use of GLC in test?
I don't know what the GLC is. Can you explain it, Dr. Arunachalam?
Hello.
Dr. Arunachalam, can you unmute and then ask me what is a GLC means? Sorry, I don't know what is GLC means.
Okay, sir.
Yeah, you can go next question, please.
Well, let's move on. Yeah.
Yeah.
So there is another question in a chat box. It's from Anandi. Is LPP is a genetic disease? Is it cancer type?
You can just check it out in the Google or a medical database. I'm not the medical biologist. I'm not oncologist, sorry. Dr. Bora Murthy is oncologist, cancer biologist, not me. I'm actually a plant biologist.
Yeah, let's.
Arunachalam sir is replied as liquid is then. This the chromatography, sir. Gas liquid chromatography.
No. The gas liquid chromatography is for analyzing compounds, right? Like small molecules.
Yeah.
The liquid chromatography, you can combine with mass spec, you can identify proteins, but that nothing to do with CRISPR.
Okay, sir.
So there is another one question. Can Parkinson be treated by CRISPR?
That's a great question. Yes, Parkinson can be treated by CRISPR. Well, it's for you. Younger generation has to find out. No, actually, Parkinson, I don't think it's a single genic mutation. Still, there's a lot of genetic understanding required to fully understand the Parkinson. So at this point now, they can easily able to fix one gene or two genes, but maybe in the future you can able to fix multiple genes. So yeah, it's possible in the future.
Okay, sir.
So I have another question here. So you told about Down syndrome, no, sir?
Yes.
In that slide, you told that genetic disorder can be found by birth. You told, no, sir. But for example, just an idea. It's not a question. Don't consider it as a question. Just I want an idea. Parent who was pregnant long back the baby was 3 months in a walk. That time when we tested, she has some genetic disorders there in that baby condition. So you may abort like that, they told, sir. But after a few days, when we take the same test, the baby does not have any such syndromes. So you can carry out in your pregnancy was the result. But when the baby was born, the baby was having some problem in his eyesight. That means when he was born itself, he was having a cataract problem. And my question is, in the first test, they told that there is some syndrome problem. But in the second one, there is no, you can proceed with the pregnancy. But the child, when he was born, he was having some problem in his body. But genetically, no one in his family have the same problem or any history, any such history was not there left, sir. My question is that how we can judge or specify that we want it or not?
Yeah, this is actually a very practical question. Thanks for asking. It is one of the fantastic questions. That is, any test, right? If you take any test, there is a potential for false positive and false negative, right? So when a mother is in pregnancy, even if you take a NIPT test, if you get the positive sign, nothing wrong in taking one more time, testing it again, right? One thing. Second thing is, if you have NIPT positive results, and it is not a bad idea, go next level of testing, like amniotic fluid. Directly, it's drawn from the ovary, like what is the thing? You can do a little further testing to confirm that one. So I would say, do not just go with one test. Do it follow-up test, and then make sure talk to your gynecologist, and is that good for or not. And then when you say the baby born and then had some kind of problem is, nowadays there are multiple tests is available, like a gene level test for identifying some kind of syndromes. I don't know whether for cataract they have a specific test or not, but I'm sure it will be in the future. And also the newborn baby, if you test with blood, certainly it will tell you, exome sequencing will tell you what the problem is.
Okay. Sir, continuous, it's nothing. If that baby grow up, when he is going to give a birth of a child or not, whether he will have the same problem, sir, is it possible?
Yes. If the baby has a problem, the thing is like there are two types of mutations, right? One is germline mutation and somatic mutation. And somatic mutations are not inherited, right? Those are the cancers kind of stuff. But if the baby developed the disorder as soon as within a few months, I'm sure it is germline mutation. As long as it is a germline mutation, it will be inherited to the next generation. So it is important to test it and then do proper advice. Yes.
Okay. Thank you, sir. Very, very nice and clear presentation, sir. Very much. Thank you, sir. Thank you.
You're welcome.
Thank you, sir.
Sir, can you stop your sharing and on your video, sir?
1 second. Now, who is doing all the thing? Like a doodling.
Somebody scribbling on the slides.
Yeah, a very large turnout, and I was surprised. I hope it's useful, and I hope I inspired 1 or 2 people at least.
Please on your video, sir. They are unable to see me.
Sorry?
Switch on your video, sir.
Oh, my video is on.
No, it's not.
Okay. I can see my video.
On the behind video.
Okay. No, I do see myself.
Many of the participants want to see you. See your face.
A participant want to see me?
Yeah,
Hold on,
they are
1
chatting
second.
in the chat box.
Okay, I will stop the video. Okay. And then I will start it.
Yeah. Now it's working.
Now you can see.
Thank you.
Yes.
So Dr. N. Kirtika has raised her hands for some question, but what I don't know what was the question, but she's raising her hands for a long time.
Okay, Kritika,
Yes,
you can.
Dr. Kartika can.
She can unmute her mic.
Yeah, you can unmute your mic and ask the question, please. Okay, there's no reply from her.
Okay, if there is no more question, I would like to thank you all, and I especially thank the principal madam and then Agila madam for the opportunity. Thank you, Ilamardi, to invite me. Appreciate it.
Okay.
Thank you, Agila madam.
Thank you, sir, for answering the questions patiently. Now,
Sir, this is Akila. Sir, you have been talking about the sequencing. See, when I find the sequence of events that is happening in your thing, from an innocent and ignorant schoolboy from Tiruvannamalai, jumping over to Annamalai University and then moving on as an enthusiastic research scholar from CAS Botany, and then moving on to US. This is a tremendous sequence of events in your life, and I appreciate very much it. And this should be a strong motivating force for all our students. Thank you. Thank you so much, Dr. Irmalai.
Thank you, madam.
Thank you.
thank you, sir.
All right.
Now I call upon Dr. Shamla Gauri, Assistant Professor, Department of Botany, to deliver the vote of thanks.
Good morning to one and all. It's my privilege to propose the vote of thanks on this remarkable day. First and foremost, I like to thank God for showering His blessings on us for today's session. I express my sincere gratitude to the principal in charge and faculty members of Pachaiyappa College. A quote by Oscar Wilde: "The smallest act of kindness is worth more than the grandest intention." This is the proudest day in the history of our college. Today, we have hosted the international webinar with more than 310 participating colleges and universities across all streams of education, from science and technology to research. On behalf of our department, I extend my thanks to our principal in charge for presiding over this function. I thank our HOD for her earnest effort in accomplishing this function seamlessly organized. I acknowledge the unwavering support received from the Deputy HOD, Dr. S. Murugesan. I express my gratitude to Dr. S. Saravanan, Senior Faculty Member, for his guidance. My heart is filled with happiness and gratitude because of the grand initiation of webinar by one of the most best-known academician in the field of botany. I would also like to express my sincere gratitude to our resource person, Dr. Rangasamy Elumalai, for giving such a valuable ideas and inspiring lecture, which will ignite the mind of young researchers to promote innovative thinking and new ideas. This will definitely help us in our studies and near future. Once again, I would like to thank Dr. Rangasamy Elumalai for taking out time from his busy schedule and enlightening us with knowledge. Sir, your talk was live and inspiring. I would also like to thank the organizing team for their support. I take this opportunity to thank all my colleagues who have extended a great support for the success of this webinar. Our profound thanks to our active participants. A special thanks to all the faculty members, participants, both from our college and other colleges. You as a participants are thanked for all your great scientific input and for many fruitful discussion and scientific interaction. One of the participants has said more webinars are needed from the resource person. Many have appreciated that the webinar is useful and informative and updated. Special thanks to Professor Bhuvargavadi Murthy, Head Biochemistry Department, IBMS Madras University for joining with all students. And also thank to Dr. Radha Venkateshan for joining with her team from Madras Diabetic Research Foundation. My special thanks to Dr. Sivamurugan, Assistant Professor, PG and Research Department of Chemistry for his technical support and guidance. I hope this webinar will serve as baseline for the research scholar and students for their expansion, not only in the academic, but also in research career. This was great meeting. Many of you have said to me, thank you for being so positive. Thank you all.
Thank you, Dr. Shyamala Gauri, for your charming words. Dear participants, the link for the feedback form has been posted in the chat box. Kindly fill it without any typographical errors. Now the session has completed to its end. Thank you for your participation. Have a nice day.
Catch you short.
Thank you all.
Thank you. Thank you all.
Thank you. Thank you.